first of all, congratulations to your paper, which is going to be published today in the renowned journal Science. Doesn't happen every day. Congratulations.
Thank you very much.
What you did was you looked inside the tumor, which is different from, a tumor is different in many respects, but inside a tumor, the cells are very dense, it's very packed, it's very uncomfortable because the tumor is fast growing, and that makes it somewhat different for cells to survive.
That's exactly what is going on. You know, when you find yourself in a crowd of people, and when it's too much for you, you have some sensors on your skin. You have also your eyes that provide the information about what is going on in your surroundings. And when it's too uncomfortable, the brain processes this information and then generates the signal that, okay, you have to leave when it's too uncomfortable for you, when it's even becoming painful, when the density is so high and your neighbors are pushing on you. So it looks like cells also have this sort of sense of personal space. And when this personal space is violated, they do need to generate some sort of behavioral output. And what was known before is that there are special sensors sitting on the cell surface, and then they can provide this information about mechanical environment of the cell. What is interesting is we found that instead of receptors or mechanosensors sitting on the cell surface, there are also mechanosensors present in the nucleus, which is the largest and stiffest internal compartment of the cell. So, and when the compression from outside is such that the nucleus undergoes deformation, there are these special molecular sensors that could detect this physical deformation of the nucleus. And the sensors become activated, and it's them who then initiate a special signaling program that tells the cell that you need to escape. And this is what we describe in the paper.
So it's good for the tumor. The tumor has an opportunity to escape, but it's bad for us because it means that the tumor is spreading.
Tumor cells, they're incredibly smart. So they have this brain in the form of mechanosensitive nucleus. And previously, the nucleus was considered as a storehouse for genetic information. And most of the studies in the field were focused on understanding how the nucleus can protect the genome, how the nucleus helps to arrange the genome in 3D, because it's a 3D structure, and depending on how genes are positioned, different cells could have different genetic programs working to kind of guide their behavior. But what we found is that there are also non-genetic functions for this organelle. So it looks like there's lots of signaling processes going on its surface, and the signaling processes are sensitive to the external mechanical environment of the cell. So cells can use it as a sort of brain, and cancer cells are incredibly smart. So they evolve.
Yeah.
They have evolved this special program that enables their escape and survival in such harsh conditions, such as the growing tumor.
But then again, you're smart as well, because we can cut into this process. We might be able to stop the tumor from growing if we use the signals and somehow block them.
Exactly. So this is where we see a potential translational element in our work, because thanks to these discoveries that we are making, we are generating a list of precise molecular factors involved in this escape program. And knowing the precise molecular targets, we could then start applying special drugs that could block the activity of these molecular players, and therefore potentially prevent tumor cell escape and survival.
Do we have potential drugs that could work?
So currently there are several biopharmaceutical companies working on phase 2 clinical trials of a few inhibitors of one of the proteins that we are actually describing in the paper. There are a few research articles already published on targeting this particular protein in the context of breast cancer, for example. And what is interesting about this protein is when it's active due to the mechanical stress, so it senses the mechanical stress activates this protein, and then what it does in its active state, it produces a fatty acid called arachidonic acid. We actually consume a lot of products during our everyday life that contain this arachidonic acid. And here's a very interesting suggestion that by modifying our diet and not consuming arachidonic acid, which is omega-6 fatty acid, and consuming more food that contains omega-3 fatty acid, or even taking pills from Apotheca, omega-3 fatty acids, we could kind of also modulate the processes that go downstream of this particular molecular compound. So meaning that lifestyle could also affect tumor development as well as response to chemotherapies, because you can imagine if we would try targeting this particular protein with drugs and at the same time still consume a lot of this fatty acid, then the drug is not going to be effective. So by modifying our diet, we could actually help the drug to act efficiently. So, but so far, these are all just speculations. These are all predictions that we make based on this very basic fundamental research.
Okay, but potentially, which kind of food would you recommend and what would you cut in your diet?
So the diet that actually supplies a lot of omega-6 fatty acids is known as Western diet. So in Western diet is rich in all sorts of fat food. Fast food, definitely one of the examples. So cutting fast food off your daily diet would be a great suggestion.
Okay, and what kind of food is rich in arachidonic acid 3? Because you recommended one of them.
Omega-3 fatty acids. What actually I do on a daily basis during my breakfast, I take a pill that contains omega-3. So this way I am ensuring myself that I'm getting a daily dose of the right fatty acids.
So you do believe in your own research?
You have to. You know, when you're a scientist, sometimes you do need to try several things on your own. Yes, and then see how it works.
Well, that's good. What is the next step going to be? So after the celebrations, because your paper, this is going to be a big deal, I can imagine, for everybody. And after the celebrations, what are you going to do? What are you going to look at the next?
So after the celebrations, I think the time will be right to sit down and write a grant to support the next steps. And what I would like to describe in the grant is the role of this mechanosignaling pathway that we've discovered in exactly the survival program. Because in the paper, we focused mostly on the ability of cells to kind of escape, to move away from the constrictive microenvironment. So it's more about cell morphodynamics, cell migration capabilities, cell invasion capabilities. while where I see my next step is to understand how this molecular program can play a role in the ability of cancer cells to make a decision between survival and death. So this is where I would like to investigate things further. And yet another interesting angle is how mechanical stress cooperates with other types of stresses that tumor cells experience within tumor, because they not only experience mechanical stress. You can imagine a patient undergoing chemotherapy or targeted therapy. So there are a number of stresses that are not purely mechanical, but also sort of chemical. So, and the idea we have is that maybe mechanical state of tumor cells also prepares it to deal with other types of stresses. And we would like to test this hypothesis further.
So it could be a biomarker. It could be something when we say, well, this is we have a lot of that and that signal here, so alarms.
Certainly, pathologists since the 19th century using nuclear morphology as a predictive marker of a particular stage of tumor development. And so far, it's been mostly as a sort of passive feature that pathologists describe when they look through the microscope at histopathological sections of tumor. But what we think is that it could be also a predictive biomarker of invasive potential. It could be a predictive biomarker of cancer cell responses to drugs. And this is where we see an exciting new direction, where we could kind of now provide pathologists with functional meaning behind these nuclear deformations that they describe, because in tumors, nuclei undergo dramatic changes in their shape and size. So now we can kind of provide the information about what is the functional meaning behind this, rather than just sort of passive signature of a tumor progression.
Yeah, that sounds very exciting. We wish you all the best for your work.
Thank you. Thank you very much.
Let's celebrate now. Thank you very much. Keep us in the loop.
I appreciate that. Thank you very much. Definitely, let's keep in touch. Bye.
Thank you.